So let's actually start getting into mitosis. Your mitotic phase, we're taking one parent cell, which is what we're looking at here, okay? And we're going to produce two genetically identical daughter cells from that. So if this is a parent cell in our body, we're taking this parent cell that has 46 chromosomes and creating two daughter cells that also have 46 chromosomes. Okay, how do we do that? Well, the first thing we've got to do is go through that interphase, meet that G1 checkpoint, so that we can go through the S phase to duplicate that chromosome number so that we have enough genetic information when we go through and divide. So remember that division is the division of the nucleus is mitosis. And mitosis, the division of that nucleus, is going to happen in 4 phases: prophase, metaphase, anaphase, and telophase. How do you remember the order? PMAP. Okay, PMAP is a way to help remember that. So let's actually talk through some of the major events that happen. Prophase is going to be your first phase, and it's going to be the longest phase of cell division because here we need to do all of the final prepping to help the cell divide. First thing I need to do is take the chromatin that I've now duplicated, condense it, so turn on those histones, make them magnetic, get them to condense, and by the time we get into prophase, we can actually start seeing your genetic information. Okay, so here is an actual light microscope picture of a plant cell in mitosis, and those squiggly lines that you're seeing in there, those dark lines that you're seeing, are the actual chromosomes starting to condense. The other thing we're going to need to do is you've got these centrioles. Remember these licorice-looking things? All right, these guys are going to be responsible for organizing your spindle fibers, which are also going to start showing up. And we need to move these duplicated centrioles to opposite sides of the poles so that when those spindle fibers are able to connect to your chromosomes, we can actually use them as a mechanism to pull the chromosomes apart. The other thing we have to do here in prophase is we have to get this nuclear membrane out of the way, because I need to get to the genetic information, the chromosomes that are inside the nucleus, so that I can move them. Well, that membrane is going to be in the way if I don't get rid of it. So your nuclear membrane is going to disappear. Your nucleolus is also going to disappear. Now, we can watch all of this happen under a microscope, but we have not yet figured out where that membrane goes, where that nucleolus goes. All right, so there's a lot of things that we're going to talk about in cell division that is pretty much all we know about cell division. Now, there's certainly some more complex things, but you're going to know the basics of what every scientist out there knows. And there's still lots of questions about how certain pieces of this work, and we're only starting, say, in the last decade to start figuring out some of the more intricate pieces of this process. So prophase, longest phase, chromosomes become visible, your centrioles are moving to opposite ends of the pole, they're producing spindle fibers as a result, and those spindle fibers are going to start to attach to your centromeres on your sister chromatids. Nuclear membrane is going to disappear, nucleolus is going to disappear. You need to know all of these major events. Metaphase is going to come next, and I think if you were to look under a microscope, this is the easiest one to see. It's also the shortest phase of mitosis. And here you've got all of your spindle fibers have now attached to your chromosomes, okay, at the centrioles. They actually attach to kinetochores, which are on the outsides of your sister chromatids. So the centromere is what is holding the sister chromatids together here in the middle. Those kinetochores are on either side of the chromatid on the outside, and they are going to be what attaches to the spindle fibers, and those kinetochores are going to be what helps to guide those chromatids along the spindle fibers in the next phase. Why do I say metaphase is the easiest one to see? Because everything's lined up right in the middle of the cell. Okay, so all of your chromosomes are going to line up in the middle of the cell to get ready to split. So if you look at the plant picture here are all your chromosomes lined up on the center, and here are your centrioles on either end. Anaphase Also, I think an easy one to see. Here we're going to actually split the sister chromatids. So they were all lined up here in the middle on the last stage. Now they're going to split. Okay, so one chromatid is going to go one way, the other half of that chromosome is going to go the other way. And they're going to start moving along those spindle fibers until they reach the poles. This is one of the stages that ensures that we evenly divide the chromosomes so that each new daughter cell has a complete set. So if this was a human cell, all right, we've got 46 chromosomes going this way, and I've got 46 chromosomes going the other. If that doesn't happen, my new cells are going to end up with an abnormal number of chromosomes, and that's going to potentially spell some problems for the new cell. All right, last step of mitosis, so the division of your nucleus, is going to be telophase. Okay, at this point, we have moved those chromosomes to opposite ends of the cell. We now have two nuclei that are starting to reform, so the nuclear membrane's coming back, your nucleolus is going to come back. And each of these nuclei has 46 chromosomes within it, if we're talking a human cell. Okay, those chromosomes are going to start uncondensing, and going back to chromatin, so they're going to become less visible over time. By the time we get to the end of telophase, I have one cell that has two nuclei. Okay, so that's what you're looking at here. Now, telophase tends to overlap with cytokinesis. Remember, cytokinesis is the actual division of the cytoplasm. So if everything's going smoothly, telophase and cytokinesis really kind of happen almost at the exact same time. So as your nuclear membrane's coming back, those chromosomes are uncondensing, your cell's cytoplasm is going to start to split. Now, cytokinesis is going to look a little different depending on whether we're talking about an animal or a plant cell. Your plant cells are going to cause some complexity because they've got that cell wall in place. And that cell wall is so much more rigid that I can't just take it and bend the cell's cytoplasm to split. Okay? In your animal cells, we're going to create what we call a cleavage furrow. And that cleavage furrow is a ring of microfilaments, remember, part of your cytoskeleton. And they're going to pinch in together, sort of like taking a big ball of Play-Doh and pinching it into two new cells. Now, I can't do that with my plant cells because of the cell membrane. So instead, they're going to produce what they call a cell plate. And that cell plate takes a bunch of little vesicles full of cellulose, which is the major component to your cell wall. And we're going to take those vesicles, they're going to elongate, they're going to stick together, fuse together, and they're going to create this cell plate. Over time, that cell plate will mature into what we talk about as a new cell wall. Okay, so cytokinesis, in animal cells, they create this cleavage furrow and split the cytoplasm, making two new daughter cells. In your plants, we need to use vesicles full of cellulose to fuse to create a cell plate that will eventually become a new cell wall to create your daughter cells. So here's just a picture of sort of taking you through all the stages one more time. So here's your interphase after G2. All right, we've got your nucleolus in place, your nuclear membrane, your chromatin, your centrioles are here. Prophase, we're going to move those centrioles to opposite ends. Your nuclear membrane is going to disappear. Your chromosomes are going to condense. We're going to start attaching the spindle fibers. Metaphase, everything lines up in the middle. Anaphase, we split those sister chromatids. They're going to start moving along the spindle fibers until they reach the pole. Telophase and cytokinesis, okay, everything's going to start coming back, condensing. We're going to split the cytoplasm in cytokinesis to get new cells, which will then jump back into this and start repeating as necessary.
Hi, it's Mr. Anderson, and in this video we're going to go through the phases of mitosis. If you ask anybody what they remember from biology, invariably it's the word mitosis, but they probably couldn't identify the different phases of mitosis, and you should be able to, by the end of this video, look right here and say, oh, that's going to be telophase, or that's going to be metaphase right there. And also you should understand what's the point of mitosis. Mitosis is technically the division of the nuclei, but what we're doing is really making sure that we can make two exact daughter cells that are identical to that parent cell. And that's how you went from that fertilized egg that was the zygote that was you to the billions of cells in your body. And it's how you replace cells as they die over time. And so you should know this, that mitosis is technically part of the cell cycle. And so a cell cycle is how a cell duplicates itself. And most of the time is in this orange phase, which is called interphase. Now, what's a cell doing during that phase? During the G1 part of interphase, the cell is growing, so it's getting larger. It then enters into the S phase, or the synthesis phase. What's going on there? It's duplicating all of the DNA. And then finally, it goes into the G2 phase, where the cell continues to grow. Now, some cells don't go through this. They don't copy themselves, like nerve and muscle cells. And so they enter into what's called a G0 phase. They never divide. But if you are going to divide after the G2 phase, then you go into what's called the M phase. And that's the mitosis, where we're dividing the nuclei. What do you get if you go through this whole cell cycle? You get two cells, and each of those independently go into their own cell cycle. And so you can think of it almost like a clock. And so cells are spending various amounts of time in each of these different phases. Where are they spending most of their time? In the interphase. The cell's growing, copying the DNA, and then growing again. And so a classic lab that you do in biology is to look at cells that are undergoing mitosis. And so what we're looking at here are onion cells. And so we've dyed the chromosomes. And so what you see is that most of these cells, not much is going on. You just have the nuclei on the inside. And so they're in what's called interphase. But some of them are going through this division of the nuclei. And so this one would be in prophase, this would be anaphase, and that's metaphase. And so since they're spending less of their time there in this one picture, we're going to be represented a smaller amount. And so the big thing that you should understand is that mitosis is not individual phases. It's not just quickly changing between them, but it's a movie. And this is a pretty amazing movie. This right here is a sea urchin undergoing cell division, becoming a larva of a sea urchin and eventually an adult sea urchin. And so in this picture, in this time lapse, the yellow is going to be dyed to represent the chromosomes. And then the blue is actually the microtubules. It makes up the spindle that allows us to divide that nuclei in half. And so if you think of it like a movie, it makes more sense to go through the whole movie before we get into each of the individual phases. And so what we're going to go through are all the steps of mitosis, and we're just going to play it like a movie, and we're going to concentrate on what goes on four different structures. So let's start with the cell membrane. So if we look through the different phases during the cell membrane, just keep an eye on that. Don't look at anything else. So watch what happens to the cell membrane over time. Nothing really. And then boom, the whole thing divides in half. And so the cell is getting larger, and then it'll divide in half. So we're forming what's called a cleavage furrow in the middle, and now we have two cells. And so now let's look at a different structure. So we understand what's going on with the cell membrane. What happens to the nuclear envelope, or that membrane around the nuclei? Let's watch that. So not much is happening. It got a little bit smaller there. And then boom, that whole nuclear envelope fragments apart, and it's gone. So we don't see it anymore. And then it reforms. Once those new cells form, then we have the new nuclei forming on each side. All right, let's go through that movie again. This time, let's look at the centrosome. Now, what is a centrosome? A centrosome is going to be made up of two things, these microtubules, and those are going to form a spindle, and it's going to allow us to equally divide those chromosomes to either side. And then in the middle, you have what's called the centrioles. And we think that the centrioles organize those microtubules and organize that spindle. Now, if this were a plant cell, there'd be some differences like a cell wall, but they don't have centrioles. We think that the nuclear envelope is important in basically maintaining those microtubules as it goes through mitosis. And so let's watch what happens to those centrosomes, and it's going to happen right away. So let's watch this. So the first thing that happens is it actually replicates. And so we're going to have one centrosome, and then quickly we're going to have two. Now let's watch what happens to those centrosomes. They form microtubules between them. That kind of pushes them apart. And then each cell is going to have a centrosome in each one. So that would be replicated again, and then this thing goes over and over and over again. Now let's look at the most important thing as we switch through these slides, and that's the chromosomes themselves. So chromosomes is the genetic information, and so it's the DNA inside the nuclei. Remember that DNA is wrapped around these histone proteins, and so when we want to condense the chromosome, we just pack it more tightly and more tightly over time, and eventually it looks like this. So this is that characteristic shape of a chromosome. Generally, when the cell is in interphase, it's just going to be loose DNA doing its job. But when it replicates, then we have to make sure that it's condensed and we can equally share it. And so what's important to know is that in this chromosome, each of these sides is called a sister chromatids, and they're attached together at the centromere in the middle. So they're exact copies. Since during that S phase we copied all the DNA, each of the sister chromatids are exactly the same. Now, one other thing that's going to show up as we go through mitosis is the kinetochore. And the kinetochore is a protein that's going to be found on the center of each of those sister chromatids, and it's going to attach to the microtubules. And I'll point that out as we switch through it. And so let's look at these chromosomes now and watch them over time. So they're real loose at this point. Now we've duplicated it. So we've copied the DNA, so you can see way more chromosomes. Now they're condensing together those chromosomes in the middle. At this point, you can see that the chromosome, so the kinetochore right here, is attaching to the microtubules of that spindle. Now some of those microtubules go all the way across the cell, but you can see a lot of them are attaching to that, or attaching to the kinetochores. What happened now is that they've all lined up right across the middle. There's a straight line along the middle of the cell. Watch what happens now to those sister chromatids. They're pulling apart, and now they're going to each of their individual nuclei. You can see that they were condensed, but now they're starting to loosen up again, and now we've just got loose DNA. And so those are the big things that are going on. And so now let's go through the whole phases. We're going to add names to it, and then just kind of talk about the most important things that occur in each of those different phases. And so we'll start with interphase, which, it's technically not part of mitosis. And so let's watch what happens during interphase. And so this would be early in interphase, and now this is later. So what's one big thing that happened is that we copied the DNA. We've duplicated the DNA. And then another thing that occurred during interphase is that those centrosomes were replicated. So now we have two centrosomes and we had one just before. So now keep an eye on that cell and see what happens as we move into mitosis or prophase. So in prophase, you can see two big things happening. You can see the DNA starting to condense here, and then we're attaching those microtubules all the way across from the centrosomes. So the chromosomes condense. What else happens at this point is that the mitotic spindle is starting to form. So when we were looking back at that sea urchin video, that was that dyed blue in color. It's really organizing the division of the nuclei itself. Now let's go into what's called prometaphase. And so during prometaphase, what are the big things that happen? Well, you can see right away that that nuclear envelope is fragmenting, so it's breaking it apart. And then the other big thing that's happening at this point is that those microtubules are starting to attach to the kinetochores in the middle. Not all of them. Some of them are reaching all the way across the cell, but you know it's prometaphase if you see kind of that explosion of that nuclear envelope. What happens during metaphase? Well, you can see now that they've all lined up in the middle, and that's how I remember metaphase means meet in the middle. And so what we get is all of those lining up along what's called the metaphase plate. It's one of the straightest lines in all of nature. Let's keep an eye on those chromatids as we move into anaphase. So what's happening is that they're moving apart. And I always think of the A standing for apart. The chromatids are separating. And so we're getting equal amounts of DNA in each of those daughter cells. And then finally, we go into telophase and cytokinesis. During telophase, what we're going to form is a new nuclei on each side. The cells are elongating, and that cleavage furrow is forming. So this is that cleavage furrow right here. It's basically taking that one cell and pinching it in half. Now, if this was in a plant cell, they form what's called a cell plate, which is a new cell wall right in the middle. What are the other big things? Remember those microtubules I pointed to earlier that weren't attached to the chromosomes? They're actually elongating the cell and allowing it to divide. And then the other thing, the important thing that happens is that we're forming a new nuclei around each of those daughter chromosomes. Now, where did that nuclear envelope come from? Remember when we fragmented that nuclear envelope before, those fragments are coming back to make the two new daughter nuclei. Eventually, we will go back into interphase. Now, where each of these cells headed, they're going back into the cell cycle again. And so the way I ask my students to remember this is IPMAT-C, And so you can just write that on the top of the test. There's mnemonics to remember it, but this tends to work for me. Remember, technically, interphase is not part of mitosis. Either is cytokinesis on the end. And so you should be able to point at each of these. So if I point at this one right here, what phase is that going to be? That is anaphase. And so what's going on? You can see those chromatids moving apart. If we were to look back here, what's this going to be? That'd be prometaphase. That's where you get the explosion of those nuclear envelopes. And so that's mitosis. It makes sure that we equally divide the DNA in those daughter cells. It allows cells to replicate, made all the cells in your body, and I hope that was helpful.